Okay, you're good to go.
Perfect. Thank you very much for the warm introduction. And hi, everyone who joined us for tonight for the PyLadies Dublin event. My name is Kristina. I am a third-year PhD student at the Interdisciplinary Programme in London at UCL, University College London. And I'll be talking to you about my PhD project in which we're trying to give single cells a story. And now if you think, okay, this is really bizarre, and what does she mean? I have a subtitle of my talk, which is captures the hashtag #DeepTree, which is an analysis pipeline that we've developed in the lab to track single cell heterogeneity within cell populations using deep learning tools. And now you may be wondering, okay, how does the scientific sort of talk relate to Python and to the topic that you usually explore at these meetups? And so to give you an overview about that, I just wanted to talk to you about how we can use Python and how can Python be very helpful for all of us in trying to answer scientific questions? So Python is not just a great tool, to be a software engineer or to do a lot of data processing in terms of, the purely computational projects that you may have experienced or heard about in your previous sort of exposure to Python. But Python can be actually used in science, and it's very popular in scientific community. So hopefully this talk will give you an overview of what you can do, and obviously there is many more applications of what you can do with it, but hopefully I'll cover some of the the hottest topics in sort of Python development and how you can use it in research as well. Okay, so before I start talking about the tools that we've developed and the way that we use Python to help us answer your questions in biology, I just want to very briefly introduce the hypothesis of my PhD thesis. And if that sounds too scientific, it effectively means that I just want to talk about what am I doing and what am I trying to achieve in my studies. So it may sound very complicated at first, but what I'm trying to see is whether cell cycle duration is heritable in cancer cells. Now, simply put, what that means is that if you imagine your cell and you're about to divide, so you're going to pop into two different cells, what I want to see is whether the time that it took your mother to divide is going to be same for the daughters of those cells. So in cell populations, you have this phenomenon when you observe that some cells divide faster than the others. And I effectively want to study and see the reasons why that is. Why do some cells divide fast and some cells divide very slow? And whether this cell cycle time is heritable across multiple generations. And this is summarized in the slide. So what we're really trying to study is that we want to see whether cell cycle time is parent dependent or parent independent. So whether the cell can regulate its timing irrespectively of the parents. Now, how do we do that? How would you approach it? You might be asking. Okay, so one of the approaches that you can use is to image your cells for a very long time. So in scientific community, we use it we call it live cell imaging for time-lapse microscopy. And what that effectively means is that you take a picture of your cells growing in the culture over a very long time in certain intervals. So for our application, it would be about 4 minutes. And we take a picture of a photo of or an image of our cells under the microscope every 4 minutes for quite a long time. So a very simple and elegant setup. We just we do this for quite a while. We do this for 3 to 5 days. And what this means is that we collect a series of images that we can later analyze. And Python is actually a great tool to help us analyze those images, because effectively what we want to do is that for every little nucleus, for every for every little for every single blob that you can see in this image, what we want to know is that how that cell behaves, how many neighbors it has, how long it takes to divide, how long it's, how many children and how much progeny it will populate, and so on and so on. So to do that, we have two cell lines. One of them is colored in green, and one of them is colored in red. And if you're wondering about more of the scientific details about that, please do get in touch, and I'm more than happy to talk you through it. But what we need to know for the purposes of this presentation, this is just enough. So we want to analyze the behavior of every single cell in our slides for quite a while. Okay, so how would we do that? How would actually how would Python and how would computational tools actually help us to dissect the behaviors of the cells. Well, so okay, you may be asking, well, okay, fair enough. So you have a noisy images and you want to isolate the behaviors of every single cell. And if you're actually familiar with deep learning, which is a kind of machine learning that we use to analyze our images, this is exactly what we would do. This is the first step of our analysis pipeline that we've developed in the lab. So the first thing that you need to do is to take your noisy inputs, your noisy image of the cells, which you can see on on the left hand side of the screen. And then you can use tools in Python and using deep learning, which can actually help you to separate those individual nuclei and the individual dots, individual cells, into separate identities. So just as you see on the left side of the slide, you provide an input, which is a fluorescence channel or a very noisy microscopy image, and what you get out of it is a nice and segmented map of where the cells actually are. And you may be wondering, okay, well, I heard that deep learning and machine learning tools are actually used for very complicated problems. Well, why is this a complicated problem to start with? Why do you need to use deep learning to just circle around cells? Probably a 5-year-old child can do that. And if you're thinking that, well, one of the things that you need to realize is that the problem that we have is actually much more complicated than what it seems. To us humans, is very clear where the cells are and where the nuclei are. We could easily take a pen and circle around the cells, but for a computer this is much more difficult. And this is difficult because the values, the individual pixel values, which correspond to the individual squares in that image, are very variable. So even if you belong to a nuclei, for example, here you have a couple of bright nuclei in the center of the scrub that I'm showing you, it doesn't necessarily mean that all of the pixels in that cell will be very bright. So it will be very white in other words. And the same is true for the background. Because this is a low resolution, because this is a low resolution image, it actually means that you can really struggle with segmenting such an image using a traditional tools. And so this is why one of the steps of the pipeline that we've developed is to use deep learning and to actually help us do this automatically, also using Python. So if you're familiar with deep learning and you've actually heard about the neural networks architectures and things like that, you may actually benefit from the knowledge that this is using a unit model architecture. But if you haven't really heard about it before, don't worry about it. The only thing that you really need to remember is that when you provide a labeled image into the model, it's actually able to internalize the behavior and to internalize the shapes of the cells, and it returns a binary mask or an annotated mask of the cells. Okay, now what can you do with that? Well, the other step, or the second step of the pipeline, is to actually use this information and give us an overview of what each individual cell is doing at any one time. So what do I mean by that? Well, first of all, because you now have the coordinates, or you have the binary mask of the cells, you can now go back into the original image and you can now ask, okay, what is that cell doing? Is it dividing? Is it just hanging around? Is it migrating on a plate? Is it growing? Is it not growing? All these sort of scientific questions that you may be interested in. And this is really important because if you want to keep track of your cells and keep track of what they're doing over time, it's very important to be able to distinguish whether a cell is dividing, is going to divide, just divided, or is not dividing at all. So to do that, we implemented a CNN model. Again, if you have never heard about deep learning, do not really worry about the technicalities. So to wrap up, what it does is that it looks at the tiny image patch, a crop of a cell just around the boundaries, and we can do that because we know where it now is. And it looks at it and it classifies one of the phases of mitosis. If those words sound completely foreign to you, mitosis effectively means cell division. So what you can do using this pipeline, using this CNN model, is that you can tell for every single cell which class or how far towards or from mitosis or cell division it currently is. Now, you may be wondering, well, okay, but can you not do this, in person? Can you not label this as a human? I'm pretty sure that you can do this. You can do this by yourself. You don't need the computational model for that, or you don't need to use the complicated Python scripts and anything to do that. But one thing that I should tell you is to analyze a lot of movies that have a lot of slides, which, you know, we've been taking for a couple of days. This is a very time-consuming task. So if I wanted to click on every single cell and label what it's doing at any one time, I would still probably be analyzing the first movie I ever took. And using deep learning at these sort of models and using the computational approach to analyze the cells is a very fast and a very time-saving approach. So later on, what we can do now, combining all of the knowledge that we have, the segmentation knowledge and the classification knowledge, so using the coordinates and the states of all of the cells, we can now do something that's called single cell tracking. Now, this may sound totally foreign to you, and if it does, I sketched a little schematic for you here on the left-hand side, and this effectively illustrates what is happening to our cells over time. So as you can see, at the beginning, you have one cell, which is, you know, highlighted in this sort of magenta color. You have one cell which over time moves on the plate, and at certain point it stops moving a little bit and it splits into two. So this is what we call cell division or mitosis. And when it stops and divides into two cells, it is now not a single parent cell, but it's now two children cells. And what we need to do if we're interested in the behavior of the cells is to be able to retain that identity, to retain the trajectory of the cells, and also to be able to store the division history of every single cell. And for that, using Python and other languages, we've developed a tool, a single cell tracker, which actually allows us to do this for many cells in a plate for a pretty long time. So for a couple of days or for a couple hundreds of frames in sequence. So this is effectively on the left-hand side, you see the schematic of what this looks like, the tracking of the parent and its children. And then in the center of the slides, you hopefully see the lineage tree or the lineage information for every single cell clone. So when a cell divides, it actually gets split into two, and the children actually get split farther and farther and farther. So this is hopefully resembling a tree structure at least a little bit. And this is exactly what we do when we're trying to have a look at the cells. And I should probably stress out that here, the BTRAC algorithm, the single cell tracking algorithm, but all of the models that I already mentioned were implemented in Python or with some sort of aid of Python development. Okay, so this is just the wrapped up slide, which is, I do realize this is a very crowded slide and it has a lot going on, but this is exactly what I just told you about. It effectively summarizes our deep tree pipeline. Hopefully now you understand why we call it deep tree, because we are reconstructing lineage trees for every single cell, and those trees tend to be very deep. So they go a couple of generations in they go a couple of generations from the founder cell to the subsequent children. And what this summarizes is that you see the cell annotation step at the beginning, where we segment and we classify using the deep learning tools and various Python scripts. And then in the next step, we do the cell tracking, where we actually connect the sequential information about every single cell into a meaningful tree. So you may be asking, okay, well, what is this all good for? Or how do you actually then utilize the information from the lineage trees? And how can you actually then tell anything about the behaviors of the cells? And if you're asking the question, congratulations, that's something that I've been asking myself for quite a while now, since I started my PhD. And effectively, this slide summarizes the answer to that. So here in the center, you can see the lineage tree, which I've shown you before, and a schematic of a lineage tree of a cell that has divided over 4 to 5 times subsequently. And what we can do and use this lineage tree for is that we can now extract the single cell information and the information about the cells at every single point of time. So we now can have a look at the density of the cells, of how much the nuclei grow, how many neighbors it has, and how much it moves over time. And all of this information, it can then be used for a sort of what we call big data analysis, or sort of big data single cell analysis, to actually give us information about how cells behave in population. So hopefully you found that interesting. And if you're still a little confused and you don't really understand what I was talking about, I would really want to show you a movie which summarizes the pipeline and actually how we can use this sort of annotation pipeline in practice. So here, what I'm going to show you is that we used our pipeline for intrinsic single cell heterogeneity. And in simple words, what it means is that you will actually see that cells do behave differently. So you'll see that the orange cell, which is highlighted, it's kind of in the center of the slide, will divide very few times. The blue cell on the top will actually divide kind of like medium times, and then the green cell will actually be able to divide many, many times. And this is actually, we believe that this is how cancer arises, because there are cells which have higher capacity to divide, and that means that they have a higher likelihood or higher potential to become cancerous over time. So let me just play this super short movie. It's quite fast, so you need to follow the 3 cells, and I'll play it again for you to appreciate later on. But hopefully now you can start appreciating that the division potentials of the cells are actually very different. And this is just picking 3 clones. If we followed other 3 cells, I'm pretty sure that they would also give us some sort of difference in the behaviors and difference in their single cell capacity to divide. I'll play it again just to make sure that you had a chance to appreciate it. And what you're watching are actually the nuclei of individual cells, which we segmented and classified and tracked effectively using Python and various Python packages. And I'll play it again because it's only 12 seconds. So yeah, this is the outcome of the pipeline in practice. And this is exactly what we use the analysis pipeline for. So now, hopefully, I've demonstrated there are cells with different properties, different proliferation and division properties. And, hopefully that triggered some interest. If that was totally foreign to you, oopsie. I don't want to play that again. You've seen it already. So I'd like to thank my supervisors, Alan Lowe and Guillaume Charras, who are the brains behind this analysis pipeline and behind this project. I'd like to thank Julia for collecting the data and for doing the wet lab experiments that you had the chance to appreciate during the presentation, and the whole CELEX team. And if you're interested in knowing more, please do get in touch. I'll be honored to I'll lead you through more of the details. And thank you very much, Vicky, for having me tonight. And the word is now yours.